Hello and welcome to this webinar titled Modernizing Biopharmaceutical Manufacturing from Batch to Continuous Production, hosted by Biopharm Asia Magazine and presented by Dr. Robert Dream and Dr. Peter Levison. My name is Sri Patel and I will be the moderator. Now before we begin, I would just like to remind our viewers that this event will be hosting a live question and answer session at the end of the presentation. You can submit a question at any time throughout the presentation via the Questions tab located at the bottom of your screen, and we will go through these at the end of the presentation. We will also be running 3 voting poll questions throughout the presentation, the first of which we will run now. So if you go to your vote tab, I'm now going to make voting active for the first voting question. The first vote says, are you actively contemplating development and manufacture using continuous technologies? Please choose one of the following. Yes, active at this time, not at present, but likely to implement within 2 years, thinking about it over the long term, or no foreseeable plans or timelines. Say click on the voting poll tab and please give your answer. Just give a few more seconds for that. Okay, and I'll keep that vote open while I continue, and I will stop the vote in a couple of minutes. Let me now introduce you to our presenters. So, Robert Dream is an industry leader with 30 years of experience, including 15 years of executive leadership experience in the pharmaceutical and biotechnology industries. He led projects, improved processes and operations through operational excellence strategies and leading-edge technologies. He is a business-minded person and has an innovative knowledge and know-how of manufacturing, supply chain and logistics, risk mitigation and risk management implementations. He's experienced in therapeutic biotechnology and biological drug substance and drug products manufacturing environments, with extensive hands-on senior managerial and executive experience at world-leading organizations. He has made numerous publications and presentations. He is a registered professional engineer and an active member of the ISPE and the PDA. He is a member of the Pharmaceutical Processing Editorial Advisory Board, the Pharmaceutical Manufacturing Editorial Advisory Board, Pharmaceutical Technology Advisory Board, PDA Letter Advisory Board, and the Interfex Advisory Council. He's a member and process chair of the PDA Aging Facilities Modernization Team. He's a graduate of Illinois Institute of Technology, his BS and MS programs, and Drexel University, where he completed his PhD program. Now, Peter Levison, in his current position within Pall Life Sciences, is responsible to the biopharmaceutical business for the global product management of the continuous processing portfolio of products and process chromatography columns and systems. This embraces continuous processing technologies, including those based upon acoustic wave separation, multi-column chromatography, and single-pass TFF. He joined Pall in July 2003, but has over 20 years experience in the biopharmaceutical industry, holding positions of increasing responsibility in research and development, technical management, and product management. Dr. Levison holds a BSc Honours Class 1 in Biochemistry from the University of Manchester, Manchester, UK, and a PhD gained in the Department of Biochemistry, University of Manchester. He has an MBA awarded through the Open University Business School, Milton Keynes, UK. He is a member of various professional bodies and has the status of CChem FRSC, Chartered Chemist and Fellow of the Royal Society of Chemistry in the UK, and CBio FRSB, Chartered Biologist and Fellow of the Royal Society of Biology in the UK. I'll now end the voting there for the first question, And I would now like to invite our first presenter, Dr. Robert Dream, to join our webinar. Welcome, Dr. Dream.
Shri, thanks, and appreciate the wonderful introduction. Today, we will be speaking about modernizing biopharmaceutical manufacturing from batch to continuous production. And basically, the drug substance and drug product manufactured in the industry, if you look at it in a holistic way and try to basically put it in boxes, it depends on what kind of manufacturing you do. It's usually the unit operations are a little bit different. But in general, you got either a small molecule or whatever you want to call it, and chemical and the biologicals. Mostly what we're talking about today is the biological mammalian cell base and microbial cell base, because this is what's been sought to do continuous manufacturing on. So if you're looking at what I have on the slide, you have the drug substance box, then you have the drug product box, and you have the fill finish or the loss, basically putting it in a unit dose to go to the patient and packaging. Then you have item 4 box that is a supply chain that supply all those 4 elements. We basically manufacture and most of the time ship across continent or ship across countries or ship across states. And if you, I basically kind of use whatever that in the bottom of the slide, it says ICHQ11, the definition of the drug substance. It's chemical entities, which we are not going to talk about today, but the biotechnology called biological entities, that kind of make it uniform, so we are not calling small molecule, large molecule API. So, MIT actually, Dr. Trout, kind of generated this curve within the last few years, trying to look at what we've done to better manufacturing platforms. And up to 2005, 2006, manufacturing was traditional. And if you look at the curve, QAE, basically quality, TPT, and those terms are defined on the slides. You see basically not much, no matter what they've done, not much improved. Then the industry went into lean manufacturing, Six Sigma, whatever they can do on existing operation. And you see some improvement, but not a lot. Then firms, institutions, basically organizations start doing different things, which we call continuous manufacturing today. And the continuous manufacturing, there's an integrated continuous manufacturing, there's hybrid, there's different types, and I kind of will mention some of those in the presentation today. But you see the improvement is drastic. And the hope is by 2020, we have a lot of those operations in place. So the search for better manufacturing platform, if we look at batch manufacturing, the operation, major efficiency gains have been implemented based on what you saw from the curve. My slide's a little bit slow. Quantum leap efficiency gain are unlikely. I mean, we reach basically the maximum we can get out of that by improving yield, by improving titer, by improving a lot of things. Manufacture and ship across continent, as I described earlier. On compliance side, you get many manual checks, deviations and investigations are difficult to root cause analysis. Basically, When you have a problem, you go back and there is not a whole lot information. You have to dig into a lot of batch record, a lot of document to find what's going on. On the quality, it's the biggest problem is you rely on in-process and end-product testing. So you hold the product sometime 2 weeks to be able to release it because it takes time to test. So where do we go from here? And on the upstream side, if I am looking a fed batch system, the old system, it looked like this. It's like a conventional fed batch system. You see a lot of big tanks, a lot of large facilities. They go somewhere up to 25K liter or some 30, depends on the type of the cells we manufacturing. And then when we improved it, we came up with using single use and many other things. We came up with still a lot better than the old type, but still not really what we need. It's still batch manufacturing. In the perfusion side, we have the old system, which is what's very complicated. It goes up to somewhere around 10,000 liter. And we improved it again using a lot of technology, a lot of automation, single use, and we improved it, but still batch system. So the total basically batch manufacturing, it depending on what you're manufacturing, it has a similarity to what you see on the slide. Now, given some equipment more or less, but that's what it looked like. It's very complicated, very large, very facility, utility, environmental, basically intensive. So we want to go to something different, which we call continuous manufacturing. Develop a platform that is modular, that is flexible, that is portable, that is compact. So instead of doing a scale up, we do scale out. It's less real estate, small footprint. It's cost effective. It's optimized. Friendly on changeover, so we can do multi-product. And the one thing that is not actually in the bullet here, it is a closed system, so we can have more than one product side by side. Then is compliant, regulatory like it, you will see from my presentation, efficient, and sustainable. So how we can do that? What we need basically in here is some actions to be taken. And we need a steady state. We need high cell density, high titer. We need high productivity, volumic flow rate, because that is what is not important in batch manufacturing. Actually, is not looked at, but we need it when we want to do continuous manufacturing. Continuous flow from bioreactor to capture. So to make it non-batch, you have to continuously basically make product over and over days. And we need continuous capture system that can support the basically fermentation. The key technologies needed, we need high speed production rate, we need a low perfusion rate. Also, we need simultaneously cell separation and clarification. So we get out of that dilemma of the batch manufacturing. Then we need a periodic counter current chromatography, basically a PCC. Now there is other technologies, but they are not well basically sought because there is not much data on them. So if somebody is working on other ones, I'm not saying here you have to use only this technology, but this technology is proven today. Then the benefit you get out of it, you get reduced bioreactor size. You can use now single-use bioreactor because the volume is small. Standard size, you can basically make a standard size line that you can manufacture monoclonal antibodies and non-monoclonal antibodies. It removes the hold steps and the hold tanks, which really that's what the batch needs, and they are in a big volume. You remove the clarification operation, and you simplify the process. So what you end up with basically reduced column size and buffer usage, which is inherent in the batch manufacturing. So let's basically look at the upstream of the continuous manufacturing after we've done all that. It looked like this. You got basically a kind of medium-sized media tank. You got small-sized reactor, somewhere around 1,000 liter. And then you got a small multi-clarification columns that basically can operate with it. And I know some people run this operation up to 143 days. So it's been well operated, well done, and there is not a lot of problems with it. Now, what I'm just mentioned is done on the basically clinical platform, is not into approved product. So to get into continuous manufacturing, you need to do more than just trying to basically reduce the tank size. You need to have very good process knowledge. You have to know your process. Without knowing your process, really, you can't do a hell lot of modification on the system. You need to have a robust equipment system integration and design. You need a support of vendors, service providers, basically, to give you that and work with you to make sure, based on your cell base or microbial base, that system will work. And then you do need to have a good control with PAT and QVD. Because without hierarchy of good control, feedback, feed forward, so these systems can talk to each other, they can produce without interruption by adjusting themselves. You don't want a person basically to be involved in there to modify things. So that is a key. And if you look at all the regulatory publications and discussions, that's what they talk about. On the downstream side, so well, actually, the slide I'm going to go through is the end of the capture and the downstream. So it kind of applies to both. So let me say this slide's kind of in between. So if you have a column and you're using a single column, that's what it looks like. The green part is the capacity of the resins. The white part, basically, there is nothing get captured. So if I am loading up my column, when I reach like 10% and I have very expensive product, now you have a product breakthrough. And that product breakthrough basically will go and you lose product. There is nothing going to capture it. It's got to go in the waste. So to avoid that, I kind of cut back and load up the column somewhere around 10% or less. Now, if I am doing continuous manufacturing, I use multi-columns. Now, this is a discussion. It depends on a lot of testing being done. Some people like threes. I've seen two, and the two is really difficult to control. I am not for it, but some people done the testing and they think it works. But I think 3 or 4 is kind of the optimal to use. And whatever breaks through from the column will go to the next column, so you lose no product whatsoever. So you have a peace of mind when you take the column up to whatever loading you are happy with. And that's what the switch cycle will look like. It kind of go, basically, you take the column off, you unload it, and you bring it back on, and so forth. So looking at fully automated column switching based on PAT. So the column in the beginning will read, you got 100% coming product plus some impurities, and the UV on the outside will show you 0. Then when you are at T1, you will see basically the inlet will still show the same thing. The outlet show you somewhere around 3% and some impurities basically getting discharged on the waste side. And at T2, you go up to about 70% plus the impurities going out that get captured by the next column. So basically that is the curve what this thing gonna ride like. Now the downstream processing, either monoclonal antibodies or non-monoclonal antibodies, will look like this. That's the old one which I showed you earlier in the earlier slide. When I modify it, I go to about 3 or 4 pieces of equipment that's integrated together, and they are much simplified down to getting your drug substance in the last tank, which is, last tank is just basically the end product. So if I am looking at the current batch manufacturing, fed batch in this case, this is what I have from beginning to end. If I modify this to go to continuous manufacturing, I will reduce the size of my media tank. I will reduce the size of my bioreactor. I eliminate the clarification and the harvest steps. I will take out the clarified harvest tank. Then I reduce my capture basically column. So you see, that is the huge, if you look at facilities throughout this country or throughout this world, doesn't matter where you go, you see they are in $1 billion to build and they are huge. This now becoming much smaller, and you will see in the next few slides. So when I reduce this, it will look like that. And an IBC platform, basically a small footprint, it's closed system. That's what you have to remember, it's a closed system. So really I don't have to worry about putting it in less grade or classified area. It has high productivity cell lines. It has chemically defined media. Now I have to get media to work continuously with my cell line. It has a universal standard platform. I can use it for various proteins. I have a steady state metabolism. I do have basically closed system, as I said, so I can minimize microbial issues. I have compatible with disposable technology. Then there is no whole tank. There is continuous flow. There is high volumetric productivity. There is an integrated modular simplified protein operation. And then I have a flexible capacity. I can decrease or increase my capacity based on what I need. And I have basically no scale up needed. I can scale out. So if I want 2 units, I just basically, And the one thing is not basically on the slide. Let me explain here. If I go to the regulatory, it doesn't matter, an FDA or EMA, or whatever regulatory you're dealing with that accepts continuous manufacturing. Not everybody accepts them yet. So if I get my documentation and licensing to operate this unit, if I copy it exactly the same way, I have a simplified approval because I just copied exactly what I had and they approved it in the past. So I can double my capacity in no time. I can go to 4 times, and if I have multi-product, I can put them in the same space since they are a closed system. So going from here, the hybrid continuous manufacturing, what I talked about earlier, there is a survey was done by that document in the bottom, that's what I basically gave reference to. It says actual use of perfusion for commercial manufacture is more widespread, but bioprocessing manufacturers are becoming increasingly reticent to publicly share details about their process. And that's the reality of this new technology. So to point out, this is the information came through the survey. It's not basically hearsay. So Genzyme, Bayer, Janssen, Merck, Serrano, Novartis, and Lonza Manufacturing for Lilly. They manufacture about 19 products. Some of them are monoclonal antibodies, some of them are not. They're all protein product, and they have an annual gross basically income of 20 billion. So you can see the technology is already used in a hybrid format, not in a from seed to drug substance, but it is used, it's out there. So what is inherent in batch manufacturing? And I tried to go through this. We actually talked about this already. Require high CapEx and OpEx, consumes large real estate, requires enormous time to design, build, validate, qualify, requires large staff to operate. At the same time, it has basically the demand for capacity is going up. Local manufacturing now is kind of sought. So if you want a local manufacturing, you can't just basically take 7 years and build a huge facility. You rather to build a new facility in 1 or 2 years. Advantage of a continuous manufacturing, we almost mentioned every one of them. Has a fewer steps, no manual handling, increased safety, shorter processing time, increased efficiency, a smaller equipment and facility footprint, flexible operation, reduced inventory, lower cost, CapEx and OpEx, less work in progress material, because when you have batch manufacturing, always you have to put it aside and test it, and then basically after the quarantine and approval, then you can release it. So that is a hurdle that you don't have anymore. Smaller ecological footprint, sustainability. Basically, you got online, inline monitoring and control, increases the product assurance. Real-time release testing, which is actually now is a document in the EMA thing, and of consistent product quality going to the patient. So what basically a manufacturer further requires, that kind of needed training, operation. You need to get regulatory. Now we have harmonization in the batch manufacturing side, but we have not have any harmonization in the regulatory side. Each regulatory basically have their own nuances that you have to go through. So there are some harmonization required between regulatories, so you can manufacture the same thing under different regulatories. Leadership basically need to be trained. That is the way of the future. Inline monitoring and control requires more improvement. We need better sensor to do inline than instead of at line or online. We need to improve technology to employ basically the Industry 4.0 or the IIoT. How to handle fault conditions. That is an item that if I have some problem, there is not a whole lot of data out there that how I can handle it. So there are ways of, and as I said, we need to implement Industrial 4.0, which is now we calling Pharma 4.0, trying to be parallel with the Industrial 4.0. Thanks for listening, and I hand it back to Sherry.
Thank you very much, Dr. Dream. So just as a reminder to our listeners, if you have any questions for Dr. Dream or for Dr. Leveson, then please click on the questions tab and send them to us. We will be asking these at the end of the presentation. We will now run our second voting poll question, which I am going to make live now. And the voting poll question reads, do you currently use fed batch or perfusion bioreactors? The choices are fed batch, perfusion, both, or neither. And we'll leave this on for a second. Please click on the voting tab and please make your vote. Just another 10 seconds for that. Thank you very much, everyone. And I would now like to invite the speaker for the second half of this presentation, Dr. Peter Leveson, to present an industry perspective titled An Update on Significant Technology Advances Enabling Integrated Continuous Bioprocessing. Welcome, Dr. Leveson.
So thanks, Sri, and hello to everybody. So following on from Robert's talk, I'm going to give you an overview of the core technologies that we at Pall have exploited for continuous bioprocessing, give you a perspective on how we've looked to integrate continuous unit operations, and then finally addressing the potential challenge and opportunity of scaling up the continuous bioprocessing platforms. So the introduction and acceptance of single-use technologies, which have been around for over 20 years now, really has been the springboard to move us towards continuous bioprocessing. And on this screen, you can see some of the technologies that have been developed and introduced into the single-use arena. And these have all been leveraged by ourselves to come up with single-use flow paths which can be used in a continuous process. Now, Robert mentioned the term lean in manufacturing, and lean manufacturing has really gained importance over recent years. It was very much pioneered by Toyota in a way to reduce waste. And there's a lot of waste within a manufacturing operation. And if you can reduce waste and come up with a so-called lean approach, then you end up with a much, much higher quality and a much more productive process. And the car is an example of a lean manufacturing process, often referred to as one-piece flow, where the components of the car come together down a production line and a car drives off regularly. Imagine if you can, that the protein molecule is the piece. Then we can now move towards a lean approach from a biopharmaceutical manufacture. And if we look at these two panels here, if you look at the upper panel, which is a current batch process, and here you have cell culture to the left. Then there's a primary clarification, often using a centrifuge or a primary depth filter, a polishing depth filter. There may be some additional sterile filtration or conditioning that goes on. You'll then go through a capture chromatography process, a polishing chromatography, and then into the final UF/DF stages. And all we've done is taken this process and evolved it into a continuous one. So it really is an evolution. And all we're doing is taking the same unit operations, just doing them slightly differently. But we still retain the same buffers, the same chromatography resins, the same filters, etc. And that is shown in the bottom panel, where again we start with the bioreactor, we go through a clarification and a polishing, we go through continuous chromatography, and then into a continuous UF/DF process. There was discussion by Robert on regulatory, and people do express regulatory concerns about moving to continuous, but we do see a lot of presentations by the regulatory authorities, and this is a slide that was presented by the FDA less than a year ago. And I think what was quite an important milestone was in 2016, Janssen got approval to convert a batch process to continuous, albeit for a small molecule. So the regulators are now supporting continuous processing, and their presence is much more visible in this area. So what I've summarized on this slide are our three core technologies that we have leveraged to move towards a continuous process. So in order to do continuous clarification, we use a technology called acoustic wave separation technology, where Pall has an exclusive license from a company called Flow Design Sonics that exploits a low frequency sound wave to do a solid liquid separation. For continuous chromatography, we acquired the intellectual property assets from Tarpon Biosystems, and that provides a single-use valve cassette that enables you to perform multi-column chromatography. And the final core technology is single-pass tangential flow filtration. We have an exclusive license from SPF Innovations to produce products and practice single-pass TFF. And that enables both concentration, but also, as you'll see presently, inline diafiltration to be performed in a continuous manner. And these three core technologies enable us to put together a complete continuous process based on single-use technologies. But it's one thing talking about it, it's another thing doing it. So in this slide here, you can see an image of our continuous bioprocessing laboratory. This lab is real. It's situated in Westborough, Massachusetts. It has a footprint of 36 square meters. And if I just briefly walk you through from the left to the right, we have cell culture at the left. We have the Cadence acoustic separator, which will do the clarification. There's polishing depth filters. We then can do an optional pre-concentration of the feed to potentially improve the productivity of the protein A capture chromatography. If we move to the back of the lab, we have a viral inactivation step, followed by a second multi-column chromatography system, which does the polishing. Then we do a virus filtration, followed by the UF/DF using inline diafiltration and single-pass tangential flow filtration before the bulk drug substance passes through a final sterilizing grade filter. And In March last year, we were able to produce just over 2 grams per hour of pure mAb from this lab. And today, through integrating the unit operations together and intensifying the process, we're able to produce in excess of 100 grams of monoclonal in 24 hours. And this means that a laboratory of this size potentially can produce, in a relatively short period of time, sufficient massive product that could get you through preclinical into early stage clinical. So this is a data set on the 100 gram per day manufacture. We produced 107.2 grams of monoclonal antibody in just under 21 hours. And in terms of the quality of the product, we've got very good host cell protein clearance and very low aggregate levels. And some of this data has been presented at various conferences, and you can see various links to it from the Pall.com website. But this is all very well, but these laboratory processes need to be scaled up to a GMP environment. And we have a lot of experience in scaling up filtration, but we do need solutions to scale up both the acoustic wave separation technology and the multi-column chromatography technology. So the Cadence acoustic separator, the benchtop unit, operates at a flow rate of around 4 liters per hour. This slide here shows scalability of the Cadence acoustic chambers, up to 50 liters an hour per chamber. And by operating the chambers in parallel, we will be able to operate at flow rates of up to 350 liters an hour using the Cadence acoustic separator process system, which we will be introducing later this year. We've scaled up the continuous chromatography. So on the image on the left is the BioSMB PD. The image on the right is the Cadence BioSMB process. And this is a full-scale GMP-compliant system. We have 8-column capability on the process systems, which we believe gives you flexibility for even the higher antibody titers and higher product titers that are coming through. It is a single-use system. One of the key features is that the single-use flow path can be exchanged within 30 minutes, and the system also operates at up to 350 liters an hour, which is the same flow rate that the Cadence acoustic separator process will operate at, and will enable you to clarify and do the primary capture from a 2,000 liter bioreactor in less than 8 hours. So the question is, does it work? Well, this is data produced with our colleagues at Merck. They had a batch process which had a productivity of 16 grams of monoclonal antibody per liter of protein A per hour. They converted it onto the BioSMB PD, so the benchtop system, and were able to increase the productivity to 56 grams of antibody per liter of protein A per hour, and then scaled it up 150-fold onto the BioSMB process. Again, maintaining a productivity of 56 grams per liter of protein A per hour. And at the bottom, Merck compared the batch process to the multi-column process at scale. And in their view, by going from batch to multi-column, they would save 16 liters of protein A resin for the chromatographic run. And that offers a cost saving of the best part of $200,000, depending on the price you're paying for the protein A resin. So the process economics of continuous chromatography really, we do think, have been proven here. So this slide summarizes the data set that we got. In terms of the number of columns, we found that 5 columns was the optimal configuration. The batch process was translated to continuous and scaled up in less than 3 weeks. And we were able to demonstrate a 150-fold scale-up of the multi-column chromatography process. We are looking at virus inactivation, and this is an image of the Cadence virus inactivation skid that is currently under development to do continuous virus inactivation. The unit, we're planning to launch it at BPI in Boston in September this year, so in 2 months' time. And this will be a great enabler, we think, to do continuous virus inactivation, which has been one unit operation step, which has been perplexing a lot of users and different ideas of how it can be done. So in terms of the portfolio of products, we have available now the Cadence Acoustic Separator PD, and later this year we will introduce the Cadence Acoustic Separator process, the full-scale GMP compliant system. We have the Cadence BioSMB PD and BioSMB process systems already available, and as I mentioned, in September we plan on launching the Cadence virus inactivation skid. We have a new range of Pegasus Prime virus filters which give very high throughput and really enable continuous virus filtration. We're introducing an acoustic wave separation system for perfusion bioreactors as well, to do the clarification of the perfusion draw-off, and that system will be introduced at PD scale towards the end of this calendar year. We have single-pass TFF technologies. We have the inline concentrator and also single-pass TFF systems already available, and we have leveraged the technology to do inline diafiltration, and the Cadence inline diafiltration system again will be introduced later this year. So with that, I'll thank you for your attention, and I'll hand back over to Sri.
Thank you very much, Dr. Leveson. So at this point, we will run the final voting poll question. So I will make the final question live now. So the question is, do you see a potential benefit in considering continuous processing as an alternative approach to one or more of your existing downstream processing unit operations? The options are, yes, I do see an immediate benefit and opportunity. No, not at this time. Possibly and would like more information, or not applicable. And please can I ask, if you do vote yes, that you do see an immediate benefit and opportunity, that you could let us know via the feedback part of this webinar, which you will fill out at the end of the presentation. You let us know which downstream processing unit operation you're talking about. So that is, if you vote yes here, can you please let us know via the feedback section which downstream processing unit operation. You can do that by prefixing in your feedback vote 3 and then letting us know which downstream processing unit operation. And I'll just leave that vote up there for a little while longer. And just a few more seconds. Okay, so at this point, I would like to begin the question and answer session. So, the very first question is for Dr. Green regarding batch definition. The question is, what is batch and how do you maintain traceability from patient to process?
Well, this question actually been asked many times over from the regulatory body and been answered many times. So I try to make it as simple as possible by referring to the sections, and I basically answer it based on an FDA thing at the moment. Anybody interested in getting answers for the EMA or others, you can email me or email Sherry, and he can basically pass it on to me. I can answer it because I can't answer all the regulatories at the same time. So the FDA basically refer back to a few sections of the CFR. CFR 21.210.3 item 2 defines batch means a specific quantity of a drug or other material that is intended to have uniform character and quality within the specified limit and is produced according to a single manufacturing order during the same cycle of manufacture. So the FDA point of view here, they say in batch refers to the quantity of material and does not specify the mode of manufacture. The other section they refer back to is 21 CFR 210.3, section 10. Lot means a batch, a specific identified portion of a batch having uniform character and quality within the specified limit, or in the case of a drug product produced by continuous process. It is a specific identified amount produced in a unit time or quantity in a manner that assures it's having uniform character and quality within specified limit. So again, they say definition for both batch and lot are applicable to continuous processing. Then they refer you back to further into manufacturing on a few other sections. Laboratory determination of final specification of release. 21 CFR to 11.165A, for each batch of a drug product, there shall be appropriate laboratory determination of satisfactory conformance to final specification for the drug product, including the identity and strength of each active ingredient prior to release. Documentation of manufacturing, 21 CFR to 11.188 says, and this is basically portion of it. I'm not going to read the whole thing. It's just a portion of one paragraph. It says, batch production and control records shall be prepared for each batch of drug product produced and shall include complete information relating to the production and control of each batch. And the two more I got. Then extended investigation of unexplained discrepancies, 21 CFR to 11.192. The investigation shall extend to other batches of the same drug product and other drug products that may have been associated with the specific failure or discrepancy. A written record of the investigation shall be made and shall include the conclusion and follow-up. And the last one says recall situation. 21 CFR to 11.150B, a system by which the distribution of each lot of drug product can be readily determined to facilitate its recall if necessary. So basically the FDA stand, they saying we have all these information for you. You have no problem really trying to do continuous manufacturing to worry about batch and where the batch basically stand and how you do recall and all that stuff. So that's my answer.
Thank you very much, Dr. Dream. So the next question is for Dr. Levison. In the continuous continuous process that you showed in the lab photograph, what chromatography technique do you use for your mAb polishing step? And if you haven't, have you ever looked at or considered a continuous gel filtration polishing step?
Okay, thank you. So the second BioSMB PD system, we run two polishing steps on it. So the first step is a flow-through anion exchange step using Mustang Q, which is a membrane adsorber. It flows through that and onto a product called CMM Hypercell, which is a cation exchange mixed mode adsorbent to do the final polishing. So that's what we use for a monoclonal. But we do have customers that have run gel filtration or size exclusion chromatography on a multi-column system. So although it's something that we're not doing with the platform that we have, we know that people who are on it, and if you need more information on that, then please contact us. Thanks, Ray.
Thank you very much. The next question doesn't say who's directed to, so I'll ask you both. When you state that in a continuous process you eliminate the clarification step, are you referring to a perfusion process only?
That's a simple yes from me.
Great. And Dr. Levison, anything to add to that?
Well, yes. I mean, if you have a batch process, fed batch, then you clearly have to clarify, and the technologies that we've developed for continuous clarification using the Cadence Acoustic Separator do that. But in a perfusion process, you still have to remove cells from the draw-off, which you can do using a sort of filtration technology, or as I just mentioned in my talk, we are introducing a Cadence Acoustic Separator for perfusion, which will remove the cells. But bear in mind that the draw-off has to pass through a sterilizing grade filter before it goes onto the chromatography. You might argue that the sterile filter does clarification. It may be in higher cell density systems that you do need to do some kind of additional polishing, but it's not to the same level that you would do for a fed batch application.
Great. The next question is again for yourself, Dr. Levison. There are several suppliers of multi-column chromatography systems around with different numbers of columns. Which is the optimal configuration?
Okay. Interesting question. All right. So philosophically, you can use one column, and that's called batch. And if you use any more than one, then you will see a productivity improvement. And I think whether you have 2 columns, 3 columns, 4 columns, 5, 6, 7, 8, it will always work. It's just how efficiently. And the metric that we use is productivity, which is the mass of antibody or the mass of product produced per gram of or per liter of chromatography adsorbent per hour. And I think all the data that we've generated and recently published shows that productivity will increase with more chromatography columns, and particularly as product titers increase. And the real reason being that you have to have one column in the non-loading stage of a process. And if you get very early breakthrough, then you need to either put more columns in the loading zone to give you time for the non-loading column to go through its wash and elution, etc. Or you have to run the thing slowly. If you run it slowly, your productivity goes down because time is a factor in the productivity calculation. So, you know, we've plumbed for 8 columns because it gives you greater flexibility. Will it work with 2, 3, 4? Yes, it will, but maybe not as efficiently. So there really is no easy answer to the question.
Great. The next question is for Dr. Dream. It starts off with a statement that no driving force is there to implement the technology for existing products. There's too much already invested in existing process, such as equipment, training, filings. What is the economic breakthrough point?
Well, as long as the existing operation, and especially for large blockbuster drug product, basically, if the system operating and already the investment is done, it's very difficult to justify shutting that down and changing it over. Now, if you have an aging facility or you basically starting with a new product, it's a lot easier to justify it that, well, I am going to spend money to build a new facility to replace my aging facility. Now, during the time, if you can do the studies and make sure that it works, it's easier to sell it because there's less capital invested in it. If you starting from a clinical and you do your clinical on continuous manufacturing, again, it's easier to sell it. So depending on the situation you are in, doing the changeover is either very difficult or very easy, but it needs convincing and needs somebody to do the work to basically convince the people who putting up the capital for the company.
Thank you very much, Dr. Dream. Just a reminder to our audience, if you've got any more questions for us, then please click on the questions tab at the bottom of your screen and send them through to us. So my next question is for Dr. Levison. Do you have any recommendations on how to carry out a virus clearance study on a multi-column chromatography system?
Okay, thank you. So the approach that we've adopted and been recommending is to basically mimic a multi-column chromatography step on a single chromatography column. So what we have to do is expose one column to the same total volume of feed as it would see were it on the BioSMB system. So, if you will, each column is going to initially receive the flow through from the previous column, and then once it would switch, it would then get fresh feed coming onto it until it broke through, when it would then switch into the wash, the elution, and the regeneration step. So we would do the virus clearance basically following a sequence like that, but using one single column, but is loaded with initially breakthrough from an additional column, followed by fresh feed. I hope that answers it.
Thank you very much. Our next question is for Dr. Dream. Dr. Dream, is continuous processing amenable to a fast changeover of products in a manufacturing facility, or is it more suitable to a blockbuster type of drug where throughput or productivity is the priority?
Well, that actually does apply to both. For companies that have a lot of a small volume per year manufacturing, that if you want to go to continuous manufacturing, you need to do a changeover. So instead of having many lines, you can use basically continuous manufacturing with single use and very easily can do a changeover. So you minimize the amount of facility you have, especially since you don't use the facility all year round. The second part is if you have continuous manufacturing is great because you don't have to shut down. You can make in a smaller volume continuously and produce the same amount of product. And the changeover, what we talked about, if you actually paid attention to the presentation Dr. Levinson was talking about, you do have changeover like on the single-use items, columns, etc. And you do need changeover, so you can use fast changeover in the process as a positive thing.
Thank you very much. So it seems like all the questions that we have today. Just as a reminder to our audience, for the final voting question, if you did vote yes, and please can you leave a further answer in your feedback remarks regarding which downstream processing unit operation you were talking about, if you did answer yes. So I would like to take this opportunity now to thank our presenters, Dr. Robert Dream and Dr. Peter Leveson, for sharing their knowledge with us. Audiences can view this webinar on demand by visiting biopharma-asia.com/webinars. Thank you very much, everyone.
Thank you.
Thank you.